urea. So this is through urea cycle. Okay, how does this ammonia convert into urea? This is ammonia received from this. Can you see the structure of urea here? So urea cycle. Okay, what is the structure of urea here? You can see there are two NH2 group in urea. So understand the structure. These are very simple structure. Though I have you can see double bond here. This is not double bond. Okay, this is not double bond. This is single bond. So this is mistake. NH2. Okay, and then CO group in the middle and NH2. Remember the structure of this urea. Okay, remember the structure of this urea very carefully. NH2, CO, NH2. So this CO comes from carbon dioxide, and this NH2 actually, one of the NH2 come from oxidative deamination. And other NH2 come from aspartate. Okay, will you remember this? Tell me.
Yes, sir.
So I will ask you. So tell me. So this is what is this is what? This is what?
Urea.
Urea. So will you be able to draw the structure of urea? No?
Yes, sir.
So what is this derived from?
CO2.
CO2. And this derived from?
Oxidative deamination.
Okay, NH3 from oxidative deamination. Oxidative deamination of glutamate. And this is derived from?
Aspartate.
See, if you remember this much, urea cycle, you actually remember, you will remember urea cycle easily. Urea cycle is very easy cycle. Only 5 steps are there. Only 5 steps are there. Okay. So after this, I want you to remember few amino acid structures. So forget about this. Only remember this. And if you see, all amino acid will have 5. 1, 2, 3, 4, 5 carbon. 1, 2, 3, 4, 5 carbon. 1, 2, 3, 4, 5 carbon. Can I write them like R? Can I write them like R? Replace them like with R group. Okay, I'll replace them with R group. So you can see with this, on this R group, if I add one NH2, this becomes this ornithine. Now on this NH2 group, if I add one C and NH2 group, this becomes citrulline. I'm adding this to this. This becomes citrulline. Now, when I remove this CO group, sorry, and then add another NH2, what does this become? This become arginine. Can you see this?
Yes.
So this means, see, I'm also teaching you the structure of these amino acids. All amino acids have five carbon. All are 5 carbon. Obviously, amino acid, if it is amino, the terminal will be a group acid and this will be amino. That's it. So this is 5 carbon amino acids, residue chain. So modification you will see here only. So can you help me making this R, then add? What do I add here? Suggest me.
NH2.
So what does this become?
Ornithine.
Ornithine. Great. Ornithine. Now, so from ornithine, what do I make?
Arginine.
Ornithine in after ornithine. So if you add something over here, so okay, let me write it. So what is this? If you add here,
Is this arginine?
is this arginine? What is this thing? After ornithine, this is citrulline. This is citrulline. Ornithine has only one nitrogen. Citrulline has two nitrogen with O group. And this is what?
Arginine.
Did you get that? Will you remember this difference? Let me draw once again. To R, to guide me, okay? So what do I do?
NH2.
NH2 group. What does it become?
Ornithine.
It becomes ornithine. Okay. And then let me draw other structure, okay? So now, to guide me, what do I do now? What do I add here?
The carbon group. The
Carbon group.
Double bond O.
Double bond O, create
NH2.
NH2. What does this become?
Citrulline.
Very good. And then, can you tell me? Last.
Nitro N.
So NH, so nitrogen. Okay, nitrogen. Then
Carbon group.
so carbon group,
Double bond NH2.
double bond NH2 and H. Okay. NH2. Okay. Let me don't worry about this 2 and 1. Okay. And then
NH2.
okay. So this becomes what?
Arginine.
So can you? I will do something, a very unique kind of thing I will do. So can you see? This becomes a triangle. Can you make it a triangle? Like this. And at the terminal region, you have nitrogen. At the center, you have carbon. Can you see this structure? This structure is, you can see this is a guanidino group. I'm also teaching you the structure of arginine in this way. So you have this beautiful group present here. You can make a flower out of this group. So you put 3 petals here, 3 petals here. So very beautiful structure. You can make very beautiful structure of this. So this flower-like beautiful structure guanidino group is attached to. This is the group of arginine. Now you will see. So I will tell you one thing. So what will happen is this arginine will release this group. Okay, this group, and this will convert into arginine, this ornithine. This you can see NH2 in between. Can you see C? Tell me.
Yes, sir.
And so this will become what? NH2, CO, and NH2. So this is urea. Now after releasing of urea, this arginine converts into ornithine, and ornithine again converts into this, and this into this. So this is urea cycle structurally. Conversion of this ornithine into citrulline, citrulline into arginine, arginine into ornithine. Again, what happens? Ornithine converts into citrulline, citrulline into arginine, arginine converts into ornithine. Again, ornithine. So what happens is this is ornithine, OC, and then you can see A. OCA will convert. OCA, OCA, OCA. Ornithine, citrulline, arginine, ornithine, citrulline, arginine. So this is urea cycle. Isn't it easy?
Yes, sir.
Will you memorize this? You have to, okay, you have to what? You have to actually revise this structure. If you revise the structure, you will never forget. You will never forget. I guarantee you, you will never forget. So you can see from arginine, urea is released, and then it will become. For conversion of ornithine to citrulline, there will be addition. Okay, there will be addition because you can see there is addition. And from arginine to citrulline, there will be removal. Addition here, removal here. So this is urea cycle. I am telling you a very essential 3-step, but there will be 5-step process. There will be 2-step here. Okay, maybe then 2-step here and 1-step here. So this will become 5-step process. So let's talk about urea cycle. Now you will, I think you will understand urea cycle. Now, do you understand this structure now?
Yes, sir.
Converts into this into this, and this into OCA, 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 OCA. Okay, AOC, you can see AOC, AOC as OAC, OAC. But this should be like first A should come, then O should come. A, because A is the initial alphabet, you can see both are vowel, A, O, and then C. AOC, 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 AOC. This will go like. Okay, AOC like this. So remember this. Now let me see. So you can see first I told you what is formed. First form is first form is ornithine. Ornithine will convert into first ornithine C, OC, and then A, and this. Okay, so remember this. So ornithine will convert into citrulline. Let's see. So what happens first is, first will be formation of this type of compound from NH and CO2. This type of compound will be formed first. NH2, and this will be with phosphate. And this require very high amount of ATP, 2 ATP. It will require 2 ATP. So this is the first reaction. Synthesis of carbamoyl phosphate. This is carbamoyl. Moyl means carba plus NH2, carbamoyl phosphate. And this is a very important type of reaction. We will see this same type of reaction in synthesis of pyrimidine. Okay, pyrimidine and purine. You know, pyrimidine and purine, these are part of nucleotide. These are nucleotide base, purine base and pyrimidine base. These are base of nucleotide. Now you can see this is require ATP. ATP when it will become carbon. So this carbamoyl phosphate will add to this ornithine. This will add to ornithine here. This carbonyl phosphate will add to ornithine. I told you two steps will be there. So carbonyl phosphate will add to this. This becomes citrullin. Can you see this becomes citrullin? So this is first step, synthesis of this, and this is second step. So carbonyl phosphate will add to ornithine. Now ornithine becomes citrullin. So citrullin will come out of the mitochondria. So this you can see this two reaction happens inside the mitochondria, and rest of the reaction will happen outside the mitochondria. You can see citrullin through one transporter called ORC1 transporter, it comes out of the mitochondria. Now with mitochondria, what will happen? This beautiful amino acid. So citrullin, you know, R group. With R group there is what? So I'm going structurally because structure help you retain memory. And then this is what is ornithine with R1N, and then to that carbonyl phosphate is added. To that you can see this is added. So this is citrullin. Isn't this citrullin? Here addition of this carbonyl phosphate to ornithine becomes change it citrullin. Now this citrullin will come out of the cell. So I will draw here only. Okay. So in citrullin, when it comes out of the cell, so citrullin aspartate will react with this citrullin. Okay, aspartate will react with the citrullin. Actually, aspartate is donating nitrogen group to this. Nitrogen group to this. You can see first. So nitrogen group of aspartate will come and bind here. Here. This is aspartate. What is aspartate? 4 carbon compound, dicarboxylic acid, 2 carboxylic, and then this, and then this. So this is of aspartate. You can see aspartate will bind like this. So this becomes a different type of amine. This is also an amino acid, a hybrid type of amino acid between aspartate. This is called arginosuccinate. Arginosuccinate, because you can see nitrogen added, this becomes arginine. Isn't this become arginine? And this is a succinic acid. Succinic acid has this without any double bond, any CO group. So this becomes succinic acid. With this N group is attached, N group attached with this. This becomes argino, this succinate, arginosuccinate. So this is arginosuccinate. This is intermediate between this. And now this arginosuccinate will be actually lysed. So what removed this 4 carbon compound, but with double bond is removed. This 4 carbon compound with double bond. This is called fumarate. So fumarate will be released and arginosuccinate will form arginine. Can you see from ornithine, citrullin to arginine? Again, arginine will form ornithine, and ornithine will enter mitochondria. So can you see OCA, O, C here, both C and then A. So the pathway will be, let's forget about those things you studied. Now I will draw the pathway. Then you join the both things, CO2 plus ATP, 2 ATP. This will form carbonyl phosphate. CP, let me write CP, carbonyl phosphate, okay, 2 ATP. And this carbonyl phosphate, so ornithine to the ornithine and carbonyl phosphate to ornithine, carbonyl phosphate will join, and this form citrullin. Okay, the enzyme, first enzyme is CPS, carbonyl phosphate synthase. This is the first enzyme. Now this second enzyme is what is the second enzyme? Ornithine. To the ornithine, this carbonyl phosphate is transferred. So ornithine trans carbamylase. Ornithine trans carbamylase is second enzyme. So this is happening inside the mitochondria. So citrulline come out of the mitochondria. Citrulline. This citrulline will react with, I told you, second amino group come from aspartate. Aspartate. And then this become arginosuccinate. Net. Okay. This arginosuccinate will convert into arginine. What is removed is fumarate. So this arginine will convert into ornithine. Ornithine. So what is released is urea. Okay, this ornithine goes. So this makes a cycle. So you can see, if you see, the first step is anabolic type of reaction. Second step also fusion type of. Third step also fusion type of reaction. This is done by AASS, enzyme arginosuccinate. This is done by AS, and this is last reaction is done by A, arginase. So you can easily remember enzyme ASS, AS, and A. So three enzymes here outside the mitochondria can be remembered easily by ASS, AS, and A. Two enzymes inside you have mitochondria, you have to memorize. So this first, second, both enzymes inside the mitochondria, their fusion anabolic step, and then third step outside the mitochondria, it is also fusion, and then you can see removal. Second, last two step you can see removal of fumarate and U. So what is there? So you can see protein catabolism will result this. Now I will tell you one more thing. So where do we get this aspartate from? We actually get aspartate from the amino group of aspartate also comes from the glutamate itself through transamination. So if you see, the ultimate source of both amino group will be in urea. Ultimate source of both amino group in urea is glutamate only, but we are talking about immediate source. So one immediate source is oxidative deamination. The second immediate source is aspartate. Okay, so you have to remember these two things. Now, and this is carbon dioxide. Now what will happen is then this fumarate will produce fumarate is 4 carbon compound with double bond with CO group here. So do you know fumarate is a part of Krebs cycle?
Yes, sir.
So it will join as Krebs cycle. Okay, it will actually join Krebs cycle. And then in Krebs cycle, and this oxaloacetate is also part of Krebs cycle.
Yes, sir.
Okay, so from Krebs cycle, oxaloacetate is coming as aspartate. And then what is resulted is fumarate, that is also going to join Krebs cycle. So can you see connection between this urea cycle and Krebs cycle?
Yes.
Krebs cycle is supplying substrate as aspartate and taking again fumarate. So this is the connection between Krebs cycle and urea cycle. So defect in Krebs cycle can cause hyperammonemia because Krebs cycle and urea cycle are associated. And this particular cycle is called aspartate arginosuccinate shunt. This is a supply shunt because aspartate is going and then arginosuccinate is releasing this. So aspartate arginosuccinate shunt, AAS shunt, AAS, aspartate arginosuccinate shunt, AAS. That is good thing. So you understood this. So you have understood this very mechanism.